Yana Bromberg is an associate professor in microbiology and biochemistry at Rutgers University. She did her undergraduate degree at Stony Brook, in double majored in computational biology or computations or computer science, right? And
Computer
biology,
science.
computer
There was
science.
computational biology back then.
Right. That's right. Computer science and biology. And then she went to Columbia University for a PhD. I won't go through the story about how she came to Rutgers. It's a funny one. Um, but in any event, she has been at Rutgers for, I don't know, 10 years now?
Yeah, exactly 10 years.
Okay. And she is the leader of Theme 2.
Okay.
Okay, you're on.
All right. Thank you for the introduction, and thanks all the speakers before me, Vic and Daniel, for making it much easier to talk now. I don't actually have to explain many of the things that would have been necessary for this particular presentation. So I am leading Theme 2 in concert with Diego Ferreira and Akif Tishkan. We're trying to figure out how the complexity of nanomachines has increased in evolutionary history. So the original objectives of Theme 2 are really nicely put. One is to identify the most common metal binding structures found in proteins and establish their similarity. Number 2 was to identify the folds and peptide motifs evolved at the root of the evolutionary tree. And number 3 was to date important structural innovations that led to new metabolic functions. Now, it's a mouthful, so basically I converted that into this. So the first question was, what are the metal binding structures? The second one was, what's the first metal binding structures? And the third one, when did these appear? So sort of reducing this, I'm just going to go through where we are on the individual components of this, goals of the theme. So number one, a long time ago, those of you who have actually seen this talk before, I had talked about this method called Sahle, which is a structural homology-based method that we developed for computing fold similarity on the basis of how likely the alignments of two protein structures are going to be reflective of the actual functional similarity of the folds that we're looking at. And now, when I talk about folds, I talk about these immediate areas that Vic had talked about, these 15 angstrom spheres around the metal binding site of the protein. So what you're looking at here is this training set that we had. You are mapping sequence identity versus alignment length in the heme binding iron sulfur cluster binding spheres. And we had come up in 2014 was this cutoff of similarity of spheres being dependent on how far the centers of ligands are within the binding. So if you took two spheres and you aligned them and they were binding heme, the heme centers would need to be less than 3 angstroms away in order for the alignment to be considered correct. And what you're looking at is the each dot representing a single alignment between two spheres. The red dots represent the spheres that should be aligned, the truth, and the blue dots represent the spheres that should not be aligned. And our point was to draw a curve through this graph. There are two curves here, but the one that I would like you to concentrate on is the one in black, is the one at Sahle 0, that's supposed to separate the spheres that are red from the spheres that are blue, so that we can figure out whether the alignment is correct or not without having to look at the ligand centers. And this was important because we were going to apply this method generically to spheres that bound all kinds of ligands. So as you can see from here, the blue is actually really, most of the blue is below the black line. The gray line is just a little more stringent, so that's Sahle 0 and Sahle 5. But the idea was that we wanted to see that the blue was below the black line and the red was above the black line. And we do see that for the training set. And actually, if we looked at all the different locations that that line can be moved up, so I mentioned 0 and 5. 0 is the one that's actually pointed to by the arrow. The 5 is slightly to the right and, to the left and above the 0. What we observed is that for our training set, at the cutoff 0, we had 95% precision, which basically meant that most of the alignments that were above that cutoff were actually correct. And we had a fairly significant recall. We didn't pick up all of the alignments that we should have had, but we had over 70% of that. Now, we could have been more strict in requiring precision, so we could have moved up, but then we would have lost the alignments that we really were interested in. So what does that give us? Well, it gave us the ability to actually look at protein structures that were unrelated to the training set. So, for instance, in this particular scenario, what you are looking at is the same type of graph for non-ion binding, non-training set data. So this is things that bound ligands that were larger than an ion, but they were not including in our training set. And what you see here actually is that we don't know what the correct cutoff is for ligand centers. And what I did is highlight 2 angstroms to 5 angstroms in red to blue scale. And again, the goal is to have the blue below the curve and the red above the curve. And what you're seeing is that even though our method, that line, the black line and the gray line, hasn't actually seen these alignments before, it's actually very good at recognizing the correct alignments versus the incorrect ones. So this is for non-training, non-ion ligands, but these are still the same ligands. What we also found is that we could use different ligands. So we could apply this alignment metric to structures that bound heme versus, let's say, cobalt ions, and try to figure out whether these look similar. Now, I guess you guys see how this is relevant to what evolution would look like. So if you started with one sphere, one type of sphere, and then you had a different environment that would encourage binding of other ligands, then potentially the sphere could be reused with very slight modifications for that particular environment. And again, even at different ligands, what you're seeing is that the red is above the black line and the blue is below the black line. So this worked out fairly well across these comparisons. And then we looked at the ions, and what we observed for ions, and I pardon me for making it too small, I guess, for you guys to see, but the idea was to see if we can represent that for ion binding structures. And what we observed that actually for the majority of the ions, you could see the red above the blue, above the black line, but there was this whole bunch of blues. I don't know if you're seeing my arrow, but I'm sort of pointing to the left corner of the graph, and there in the upper left corner there's a bunch of blues, and these guys actually are all triangles which are copper. And so if I split it up into the individual ions, you could see that the only problem is here, because the blue on the top left is only here, not here, not here, and not here. And if I blow up the copper, you see that it's over here in the top left corner that you're seeing issues. So I actually went in and I explored the reason for why this happens. And this is an interesting thing in itself. But what I observed is that the PDB, which is where we got our data from, has a lot of mutagenesis studies on the movements of ions across the structure, throughout the structure. And actually, there is a bunch of the structures that have a copper specifically fixed in a different position than it would have to have been for natural activity. So what I found is actually about 80% of the structures that had this kind of weird way of binding copper had the word mutagenesis or synthetic design in their abstracts and titles. So I felt that this was okay for continuing to carry out the comparisons, because it actually just referred to the synthetic issues or PDB issues in describing the natural world. So I guess the bottom line here was that we could use the Sahle scores at 0 cutoff, at this black line cutoff, in order to be able to describe whether 2 particular spheres are actually connected in a functional way. So the 2 spheres not only look alike so that they can be structurally aligned, but they're also functionally similar. So if we took that as a reference, we could potentially align all the spheres that we had in our set. We had some 4,600 spheres. They were chosen to be very precise. They were not 100% sequence identical. They came from different structures and so on and so forth. And in the end, we were able to generate a network that connected spheres, protein spheres, to other protein spheres with a Sahle cutoff of at or below 0. And what we found is that that network looked like this. Now, at various iterations of this talk, you've seen this network look slightly different. It's just a matter of layout. But I think the main point remains in every single iteration of this network. Sorry, apologies. In every single iteration of this network, what you're seeing is that, first of all, out of all the 4,672 spheres that we had in our set, 4,613 were connected, which is already interesting. But also what we found is that the structures that bind particular ligands, so this is copper over here, all copper ligands, including the ions. And this is heme here, and this is the iron sulfur. This is the iron sulfur clusters, and the red is the iron ions. And what you see is that the hemes and the copper are on the different sides of the network, and manganese ligands are distributed all throughout the graph. What you also see is that it's very likely that the structures represented in these blobs over here may be due to just redundancy of the PDB, because if someone's interested in studying hemoglobin, for instance, they're going to determine that structure multiple times, not one person per se, but many people. At some point, I'm told that the PDB had contained 25% of hemoglobin structures, so of all the PDB. This is now a long time ago, but nevertheless, that's the idea of the redundancy that you can expect in the PDB. So in order to be actually able to figure out what's going on in this network, we needed to figure out, is there any way that we can reduce this redundancy? So one of the things that we needed to do for that is, I'm sorry, before I get into that, there's two things here. One, we validated the Sahle score. In order to validate the similarity network, we had to validate the Sahle score, and I didn't talk in detail about it, but the mutagenesis story on copper was actually part of this conversation. And one more thing that we are still working on in collaboration with Diego Ferreiro and the postdoc in my lab, Ariel Aptekman, is basically comparing our similarities that we established with Sahle versus this stylability score, which Diego had introduced a while back. So we're going to validate the connectivity, even though that we feel that Sahle scores are sufficiently informative as is. All right, so reducing the bias. In order to identify what were the first metal binding structures, we had to reduce the bias. And this is a brief summary of what the bias looks like. So if I actually asked a clustering algorithm to tell me, at Sahle score of above 0, what are the clusters that contain 100% of the same ligand binding proteins, what I would observe is that there are 1,551 clusters, which basically means that rather than having 4,613 proteins, we can choose the representative proteins per cluster and actually just rely on them rather than looking at anything else, at the entire collection, in order to be able to describe the diversity in the world. So here, a single line, for example, here represents the number of spheres that bind heme. So the first one is the ligand, the second one is the ion in that ligand. And what you're observing is actually there is a bunch of the x-axis here is just arbitrary numbering of clusters. But what you're observing here is that there is a bunch of heme clusters all around. So this dot over here, the really large one, is dot representing 50 heme spheres. And basically they're all the same. They bind heme and they have a very large Sahle score, or large enough Sahle score, which basically suggests that this is the same protein determined multiple times. So by simply taking the representative proteins, these 1,551 clusters, and actually, to be honest, it's 700 singleton proteins, which are unique, and then another 800 or so, which are clusters. So if we were to take these clusters, we could represent the network in a much smaller shape. Now, the other thing that we wanted to do, because we're doing this all for the purposes of aging proteins, is to figure out what are the ages of these proteins that we're looking at. And since it wasn't very easy to compile a database of ages for these proteins, what we decided is to map them to existing trees, bacterial trees, which actually have somewhat of an aging option there. So not an absolute age, but a relative age for these trees, for these proteins. So what we did is we mapped the protein sequences to the genomes of bacteria. So now we're looking at 143,000 sequenced genomes in this database called the Genome Taxonomy Database. Those of you who remember, originally we started with a much smaller set. By using this larger database, we were able to annotate almost all of our proteins. And what we thought to do is take the oldest appearance, so the organism that had the protein that we're interested in longest time ago as the reference date for this particular protein. And then by taking the ages of these proteins and by taking the representative clusters, cluster representative proteins, we were able to reduce our large network of 4,600 organisms to 1,509 nodes. Now, this is a reduction that is really big in terms of the number of proteins, but it's not a reduction which actually altered the network connectivity by much. So what you're seeing here is this smaller, reduced network, and I'm going to put in the corner, it's not meant to be very visually appealing, but that was the original 4,600 network. And what you're seeing is that we still have our heme groups, for instance, that used to be over here, but these are actually now a lot more representative and condensed of what it used to be. Now, we also observed that if I look at the entire set of the ages of these spheres, this is the full set, that distribution, right? So you see the highest peaks are over 800 here, but that's the distribution of the entire 4,600. And here's the representative set that I just showed you. And what we're seeing is that these distributions are pretty similar, which suggests further that the proteins that we've selected actually represent the full diversity of both the ages and the structures that would have been available in the entire full set. Now, just to sort of run ahead of myself, why we're doing this is because what we want to do is kind of like what Vic's group is doing, is basically trying to identify which components of networks keep appearing over and over again. And for that, we want to be able to say, okay, this is the limited set of spheres or folds that we should be looking at. All right. So what we didn't observe is that there was a correlation between the distances in the tree, in the evolutionary tree that we had, and the Sahle scores. So there was some correlation, but not much, right? And you could see from the distributions that you're looking at is that the distances in the tree actually are a full normal, right? Whereas the Sahle scores are more skewed towards the left. So it's you expect the correlation to be negative because a lower Sahle score indicates a higher distance in the tree, but you would expect it to be more, and it's not. And we thought about why that would be, and we came to the conclusion that actually the age of the organism to which the fold maps is probably not the correct age to date the fold. So what we did is we actually went back to the tree and we said, what we should look for is the origin of the fold. And we could theoretically say that this is at the origin of the last common ancestor. The origin of the fold is at the last common ancestor of all the organisms that carry the fold. So for instance, if there were two organisms in the acidobacteria that had a particular fold, then the origin of this fold could be mapped to over here where the purple square I had put up. Now, if the fold was also present in these organisms, then the origin would potentially be earlier. So we tried to date the least common ancestor or the last common ancestor for these particular folds. And what we observed is this. Here's on top is the normalized age, and we have these unassigned ages over here. But other than that, what we observed is that the age is actually distributed, interestingly here. But the last common ancestor age is actually very much limited to these two, right? So at the origin of this tree, this is the root at 0. And then this is a bunch of folds that fall into this group of, fall into this root, which is distributed across a large number of organisms, just not all of them. So by using the age, the LCA age and the Sahle score, we were able to actually assign weights to the individual connections of the proteins. And to further reduce the world to things that were most interesting, we asked for a minimum spanning tree for the entire set of spheres that we were looking at, which basically resulted in what you're looking at right now. So again, the weights to edges are assigned to edges on the basis of the Sahle score, the LCA age, and the age of the assigned fold. And what we observed is that actually, if you took the most central spheres in this network, you would find these, I think there's 10 or 11 of them, and they're colored. So these are copper, these are iron, this is manganese, and so on and so forth. So if you look at these structures, here they are. They fall into these organisms that we're looking at here in the middle. They're mostly oxidoreductases, which is in line with the expectation. But what I want to focus a little bit on is what does this mean in terms of dating these original folds? So we are going to take these structures and identify the subfolds of these, and this is going to be done by Ariel, and maybe he's going to tell you about this tomorrow. But what we want to do is take the identified subfolds, eventually identified subfolds, and hand them off to experimental work to Akif and to Vic, and see if we can actually do anything with them, synthesize them, and see if they make sense as origins of contemporary metalloproteins. So the third question in the goal was to ask when did the structures appear? And this is a normalized age in the tree, basically just a different shape of what you've seen before. It seems like they appear fairly early, but fairly early is not an answer to when. So one thing that potentially we could do in more detail, and this is just a basic idea, is to look at these phyla that these organisms belong to and go back to the tree. I had another slide in here, but go back to the tree and try to date the ages of these phyla. So one very important point is about this organism here, that's Endomicrobium proviatum. I was actually very excited about this organism. It's a deep branching clade of mostly uncultivated bacteria in Microbia phylum. It's an obligate intracellular endosymbiont of a termite gut flagella. So you have these matryoshka here. Basically, this thing is an intracellular symbiont of a termite symbiont. So basically 2 levels, 3 levels of symbiosis. And the idea for why that's exciting to me is because these organisms are like our mitochondria, basically. Not in the sense that they produce the energy, but in the sense that they have very minimal genomes, very small and simple proteins, and actually really probably look like something that would have been there in the very early stages of life. So I was excited about this phylum. But here's the tree of the phyla, just in case you were interested in terms of the ages of these. So this over here is the Microbial. It's the very origin. So time goes like this. So the arrow is in the middle. So this Microbial are probably at the very origin of the bacterial appearance. This is the second one. This is the proteobacteria, the geobacter that we are looking at as well, who are pretty old, right? They don't appear until this split over here. And the Firmicutes, which we also got, appear only at the end. So towards the end of this stuff. And actually, if I were to put a timeline on here, what you're seeing is that, let's say, proteobacteria appear about 3 billion years ago. Bacterial root is about 4 billion years ago. So the elusive bacteria, elusive microbiota have a root that's somewhere here between 3 billion and 4 billion years ago, which is a large span, but we're working on making it slightly more precise. Anyways, that's the story of the dating. We are working to characterize the interaction between the proteomes and their environments. Currently working on it, so you will be seeing a couple of slides, I hope, a couple of talks in the next couple of days to describe how we are doing that. We're characterizing the interaction between proteomes and their environments by correlating metagenomic functional and metal cofactor annotations. And that's work by Ariel and Adrian Horfrost in my lab, and Joy Bongiorno in Team 3, Shauna Morrison, Donato Giovanelli, and Bob Hazen. And then there is this picture that we had to build the machine learning tools to predict molecular functions present in the given environment. And again, it's Adrian and Theme 3 people, but also I'm looking for a person, actually, if your friends are computationally inclined and looking for a position, we're looking for a PhD student or a postdoc to take this on as well. So that's it. That's the story. I showed this image last time and I still feel this way. I feel like there is these two really awesome things going on either side of me, and I'm just trying to bridge the two of them together. And thank you for listening.
All right, thank you very much, Yana. That was, you're between a rock and a hard place, I guess.
I wasn't gonna say it, but